fertilization from ejaculation. Before I get started, I just want to mention though that whenever I refer to insemination, I am referring to natural insemination as opposed to artificial insemination. Also, there are some printouts on your desks in front of you. There's also a color-coded note card with a number written on it. On the printout, there is it's separated by colors. So I'm going to call on a color, and if you could just read the number on your note card that's correlating to the number that's on the printout. Makes sense?
Yes.
Okay. So for starters, who has seen the movie Look Who's Talking? It's starring John Travolta and Kirstie Alley. And it has Bruce Willis as the voice of Baby Mikey. Nobody? Okay, cool. When I was a kid, I just really loved this movie so much because in the beginning it shows this scene. It's called the sperm rings, and it always made me feel like I knew exactly how babies were made. So, that looks legit, right? And I'm guessing this part's the cervix, because there's like a portion where they dock in the cervix, so I'll talk about it later. That's cute, right? So it's honestly like a fairly accurate depiction of fertilization. But today, I'm going to talk about it a little bit more in detail. So we're going to discuss the major sequence of events following deposition of the spermatozoa into the female reproductive tract, leading up to fertilization. So spermatozoa are exposed to a series of different environments that significantly alter their numbers and their function. And so we will discuss immediate transport and then transport through privileged pathways in the cervix, transport through the uterus and the oviduct here, which is actually the site where the oocyte and the sperm meet and fertilization takes place. All right, so this is just the image before, just a little bit bigger so you can see more clear, and I'm kind of just going to briefly explain everything before I jump into it all. So, following copulation or insemination, seminal fluid containing sperm, also known as semen, is deposited into the female reproductive tract. After their deposition, the spermatozoa are lost from the tract by retrograde transport, as you see falling back here, or by phagocytosis by leukocytes. The remaining spermatozoa must migrate the cervix, as I mentioned, through these privileged pathways. They have to enter and travel through the uterus, and finally into the oviducts when they will encounter the egg and fertilization.
Question.
Yes.
You mentioned the retrograde. Can you go a little more detail? What is it?
I'm going to go a little more detail further in that. This was just a brief overview.
Okay.
So, semen deposition. In some animals, the male ejaculates into the cranial vagina, as in the cow, the sheep, the rabbit, us, primates, dogs, and cats. And in other animals, semen is deposited directly into the cervix. In the pig, or is squirted through the cranial lumen, as in the horse and camelids. In the dog, pig, and the horse, most of the ejaculate gains entrance into the lumen, but in the cow and the ewe, almost 60% of the spermatozoa is lost to the exterior through retrograde transport that I just mentioned. So, retrograde transport is semen that just naturally flows toward the exterior of the vagina, toward the vulva, instead of going forward toward or through the cervix. The degree to which the sperm are lost from the female tract depends on the physical nature of the ejaculate, as well as the site of the seminal deposition, and I'll go a little bit further into that in a second. In some species, the seminal plasma contains coagulating proteins that form a conspicuous vaginal plug to prevent the sperm from undergoing this retrograde flow to the exterior. So the stallion ejaculates in a series of jets in which a sperm-rich fraction is ejaculated first in 3 to 4 high-pressure squirts. This fraction contains about 80% of the spermatozoa, and the last 5 to 8 jets are of lower pressure, and they contain fewer sperm. The seminal plasma in the final jets is highly viscous or thick and may serve to minimize retrograde sperm loss from the marriage tract. Now, the boar ejaculates 200 to 400 milliliters of semen, and because of this large volume, most of the semen flows from the cervix into the uterine lumen. As in the stallion, the boar also ejaculates in a series of seminal fractions. The first fraction consists of excessive fluids and a gelatinous coagulum, but few sperm. The second fraction is sperm-rich, and this fraction is followed by another, which forms an additional gelatinous coagulum that actually resembles rice pudding. And that's used, of course, to minimize that retrograde loss. In the dog, semen is ejaculated in 3 fractions. So the first is a pre-sperm fraction that's thought to originate from the prostate, and it has a really small volume ranging from 0.5 milliliters to 5 milliliters. The second fraction is sperm-rich. It's between 1 and 4 milliliters and contains 300 million to 2 billion sperm. And then this third fraction ranges in volume from 1 to 80 milliliters, and because of the tie that Joy actually mentioned, it's believed to push this sperm-rich fraction ahead into the uterus. And now, earlier I also mentioned that another way that sperm are lost from the female tract is by phagocytosis, by neutrophils. So when the female reproductive tract is under the influence of estradiol during estrus, neutrophils, which are powerful phagocytic white blood cells, sequester in the mucosa of the tract, especially in the vagina and the uterus. These neutrophils are poised there to attack foreign materials that are introduced into the tract during copulation or insemination. And technically, spermatozoa are foreign to the female tract, right? And so just one neutrophil is going to be capable of engulfing multiple sperm, and they don't differentiate between live and dead sperm. And so in the figure here, we can see 3 different leukocytes labeled A, B, and C. Inside of the leukocyte, you can see sperm heads that they are digesting in their cytoplasm. And then coming from leukocyte C, you can also see a sperm tail here. So leukocyte infiltration. Studies have shown that within 6 to 12 hours after the introduction of sperm into the uterus, there is a large migration of neutrophils from the uterine mucosa to the uterine lumen. While leukocyte infiltration is an important contributor to post-insemination spermatozoa losses, this infiltration is also important to prevent reproductive tract infection. So let's check in. For those of y'all who just got here, there are some handouts in front of you and a note card. On that note card, there's a number. Right now, I'm going to ask that everyone who has a blue note card read the corresponding number on the handout, please.
So after deposition, spermatozoa are lost from the reproductive tract versus binutrients. However, factors that regulate loss of sperm are among the least understood phenomena in reproductive physiology.
To relate the ability of the female to retain viable sperm may influence fertility of the given baby.
Thank you. So, following deposition, spermatozoa must navigate through the highly convoluted system of grooves within the cervix. And this here is actually the cervix of a cow or of a ewe. As we learned in other presentations, the cervix of different species of animals look different. So let's just mention that. During estrus, however, the cervix produces two different types of mucus. One is called the sialomucin, and the second is the sulfomucin. And the surface also acts as a filter that eliminates non-volatile sperm. So cervical mucus. The sialomucin is a mucus of low viscosity. It's produced by cells in the areas of the cervical crypts. So it's these spaces down here. And then the sulfomucin is produced by the apical portions of the cervical epithelium, covering the tips here. And it's actually quite viscous. It's like really thick, kind of like syrup or molasses, I guess, right? So this is just that same picture, a little bit blown up so you can see it a little better. The production of the two types of mucus, one of low viscosity and one of high viscosity, creates two distinct environments within the cervix. And so spermatozoa that are encountering the viscous sulfomucin up here are going to be washed out of the tract, while those that encounter the low viscosity sialomucin down here are going to be able to actually swim through these crypts, through these privileged pathways, and actually enter the cervix. So, as we already know, estradiol is high during the follicular phase when insemination occurs. And estradiol stimulates contractions of the muscularis, particularly the myometrium, and this aids in spermatozoal transport. Also, prostaglandins in semen, like PGF2 alpha and PGE1, cause increased tone and motility of the uterus and or the oviduct. Transport of sperm following copulation can be divided into two phases, and that's the rapid phase and the sustained phase. So, during the rapid phase, within a few minutes after copulation, sperm can be found in the oviduct already. And so the rapid phase of transport was once considered to be important because it delivered the sperm to the site of fertilization very quickly after copulation. And they postured themselves there and wait for the oocytes to come after ovulation. However, after further research, it was shown that the sperm that actually arrive in the oviduct within those minutes after copulation are not even valuable, so we can't use them. So the functional importance of this rapid phase is actually still not obvious, but it's believed to just simply represent a burst of transport that's due to the contraction of the muscularis. Yes?
So when you say, we might be getting to this next, but like how you said, like within a few minutes, it's sperm or spermatozoa that's found in the oviduct. How long does it take for a sperm to go to the oviduct that's
It normally takes a couple of hours. It's a long process because the sperm has to go through full maturation. I know that sperm matures in epididymal, but it doesn't fully mature and become fertilized until it's in the female reproductive tract for a minimum period of time. I do go into it a little bit later, but thank you, it's a good question. So the more important component of transport is the sustained phase, in which sperm are transported in a trickle-like effect from the so-called reservoirs in the cervix that I had mentioned earlier, and the uterotubule junction. So during the sustained transport phase, sperm move into the isthmus and attach to the oviductal epithelium, and I'm going to go into further detail and show y'all exactly what's happening there. They can actually attach to the epithelium throughout the entire oviduct, but they attach near the uterotubule junction because this is the first oviductal region that they encounter. And this docking is crucial to sperm survival because it elicits a signal cascade in sperm that promotes their viability. And then without docking, sperm will die within 6 to 12 hours after insemination. So that kind of goes into the question that you asked.
Yeah.
So, as you can recall from chapter 3, sperm will acquire maturity, like I just mentioned, during epididymal transit. However, the maturational changes that occur in the epididymis don't render the sperm completely fertile. For maximum fertility to be achieved, the sperm must reside in the female reproductive tract for a minimum period of time. And during that time, they'll actually undergo changes that allow them to become fertile. And these changes are referred to as spermatozoal capacitation. So, the site for capacitation varies among species. In species where sperm are deposited into the cranial vagina, capacitation may begin as sperm ascends and passes through the cervix, as in the bitch here. In species where semen is deposited into the mid-cervix, like the sow, or into the caudal cervix, like the mare, and it immediately enters the uterus, capacitation is probably initiated within the uterus and then completed in the isthmus of the oviduct, as is the case with all species. But an important concept to remember with regard to capacitation is that the process can actually be reversed by returning capacitated sperm back to the seminal plasma. And this process is actually referred to as decapacitation. So this is just a conceptual version of mammalian capacitation, and I'm just going to briefly go through it. So the plasma membrane of the epididymal sperm contains a complement surface of molecules like proteins and carbohydrates that can be seen as these yellow T's here. The surface molecules in the epididymal sperm become coated with seminal plasma proteins, which are these orange halos around the T's now. And these seminal plasma proteins mask the proteins of the membrane. And when the sperm are exposed to the female reproductive tract environment, these seminal plasma coatings, along with portions of those surface molecules, are actually removed. And then we will have portions of the molecules on the sperm here that can actually bind to the zona pellucida of the oocyte. And I'll go further into detail in a second. So now that the spermatozoa are fully fertilized, it's important to note that copulation has to take place within a few days of ovulation. In most domestic species, the period of estrus is less than 24 hours, and so ovulation only takes place within a few of those hours. Whereas in contrast, the fertile lifespan of sperm varies in different species, as you can see here. So, for instance, in the woman, if you were to, let's say, have sex with your boyfriend on Monday, and you don't actually ovulate until Wednesday or Thursday, you can still actually become pregnant. That egg can still be fertilized because those fertile sperm are just going to be sitting there waiting in the reproductive tract for the oocyte to actually reach the point of fertilization. So, once ovulation occurs, the oocyte is going to enter the oviduct through the infundibulum here, which I think Erin explained to us. Tiny cilia that I don't know if y'all can see very well, but it's all along the inside of the oviduct there, push the oocyte toward the ampulla, which right here you can see this is where gamete interaction will take place. As capacitation is completed in the isthmus, which is this part of the oviduct, the spermatozoa will actually become hyperactive, and I'll get into that in a second. But the hyperactivity will help them move through the isthmus to the site of fertilization here at the ampulla. So, yes, ma'am?
So, is it possible for, I know you like to say that the egg and sperm meet in ampulla, is it possible for them to be in the isthmus?
No, it's not actually possible for them to be in the isthmus because in some species, there's actually like a junction here that doesn't allow unfertilized eggs to pass through. So they have to meet in the ampulla in order for fertilization to occur.
What's that area called?
I'm sorry?
What's that area called?
can't remember right now. It's the ischic junction, I want to say. Ambulary ischic junction. Okay. Yeah. So, hyperactive motility is when the motility patterns of sperm actually become hyperactive and change from a progressive linear motion, where they swim in a relatively straight line, kind of like an Olympic swimmer, to a frenzied dancing motion that's not linear and is localized in a small area, kind of like a disco dancer. And so it occurs throughout the oviducts, and like I mentioned, it's brought about there by specific molecules that are produced by the epithelium within the oviduct. And hyperactivity is thought to actually facilitate sperm and oocyte contact. So let's check in. If you have a purple note card in front of you, can you read the corresponding number on the handout? And this is just to kind of go over what we have gone through so far.
Spermatozoa must reside in the female reproductive tract before they acquire maximum fertility capacitation, which is completed in isthmus. Copulation must take place within a time period that is close to ovulation to ensure successful fertilization.
And hyperactivity takes place after
Capacitation.
Capacitation is completed to facilitate sperm oocyte contact.
Thank you. So, like I mentioned, the sperm are known to contain these specific proteins on their membrane that enable them to bind to the zona pellucida proteins. The zona pellucida of the oocyte actually consists of 3 glycoproteins, the ZP1, ZP2, and ZP3. Zona proteins 1 and 2 are structural, while 3 is much like a receptor for a hormone. So, this is just a model for zona binding and the initiation of the acrosome reaction in mammalian sperm. You can see this purple is the sperm plasma membrane, and then this pink area here is going to be the zona pellucida membrane, and then the white space there is going to be the perigonadine space, and I'm going to go a little bit further into detail about that. But the sperm plasma membrane that's overlying the acrosome contains 2 receptor-like regions that you can see here, and then this is the acrosome space. The first region is called the zona binding region, and it reacts with the ZP3 protein on the zona pellucida membrane to cause physical attachment of the sperm to the zona pellucida. The second membrane region is called the acrosome reaction promoting region, and that's this region here. And that region also binds to the ZP3 protein on the zona pellucida. And this reaction is, it causes the sperm plasma membrane to fuse with the outer acrosomal membrane. And that's what's shown here by these arrows. So the purpose of the acrosomal reaction is twofold. First, the reaction enables sperm to penetrate the zona pellucida. And second, it modifies the equatorial segment so that it can later fuse with the plasma membrane of the oocyte. The acrosomal reaction begins with the plasma membrane of the sperm and forms the fusion sites within the outer acrosomal membrane. This process is called vesiculation. So, before the acrosomal reaction begins, all the membranes are intact, as you can see here. So, we have the plasma membrane, that's this clear area. The outer acrosomal membrane is the space that's next to that. You'll have the acrosomal contents, which is what's in the middle there. And then the inner acrosomal membrane is going to be the darker color. We have the postnuclear cap here. And then this sphere-looking thing, that gray thing there, is the actual nucleus of the sperm. So during the reaction, the plasma membrane that's overlying the acrosomal membrane begins to fuse with the outer acrosomal membrane. And the fusion of the 2 membranes leads to vesiculation here, which creates pores through which the acrosomal enzymes that are in the acrosome content can pass through. And this allows the sperm to actually penetrate the zona pellucida. After the reaction, the vesicles are sloughed off, and it leaves the inner acrosomal membrane, the equatorial segment here, which is where you see these fusion proteins, and the post-nuclear cap is still intact as well. So penetration of the zona polluta by spermatozoon is believed to be a rapid process that probably takes no more than a few minutes. Following attachment to the zona pellucida, the acrosome reaction allows the release of a variety of enzymes, one being acrosin and its inactive form proacrosin, which I'll explain in just a second. But the mechanical force that's generated by the flagellar action of the tail may actually be sufficient to maintain that sperm head contact with the zona pellucida so that the sperm can actually digest that hole through which it can pass. And so, I just mentioned, acrosin is released during the acrosome reaction, and it hydrolyzes the zona proteins and enhances the sperm's ability to digest into the zona pellucida. In the inactive form, acrosin is known as proacrosin, and it has a strong affinity for the zona and thus aids in binding. And so as proacrosin is converted to acrosin, the sperm begins to penetrate and actually make its way through. So let's check in again. If you have the green side notes, can you please read the corresponding number?
5 to the zona acquires specific zona binding proteins on the spermatozoal membrane.
The acrosomal reaction is an orderly fusion of the spermatozoal plasma membrane and the outer acrosomal membrane.
Release of the acrosomal enzymes like acrosin allows the sperm to digest its way through the zona pellucida.
Great, thank you. So finally, we have reached sperm and oocyte fusion. And I blow this picture up, these images up, just so that you can see them a little bit better. So here's the first one. When the spermatozoon completely enters the zona pellucida, it reaches the perivitelline space, like I mentioned, that white space here, and it settles into a bed of microvilli that are formed by the oocyte plasma membrane. And so the actual fusion of the oocyte plasma membrane and the equatorial segment, which is here, I mentioned earlier those fusion proteins that are on that portion of the membrane, is believed to be the fusion of the oocyte plasma membrane and that equatorial segment is actually believed to be brought about by those fusion proteins that are located there. And prior to the acrosome reaction, those proteins are inactive, but once activated, they enable the sperm membrane to fuse with the oocyte membrane, and then the sperm will be engulfed. And I just wanted to point out here that you can see these cortical granules that are created in the cytoplasm of the oocyte. They are, during this period of time, before the actual fusion, they are moving to the outside of the oocyte membrane. Once they migrate to the periphery of the oocyte, they will actually undergo the cortical reaction, which is when the cortical granules undergo exocytosis and their contents are released into the previtelline space. This results in the zona block as well as the vitelline block. So exocytosis, like I said, results in the zona block, which is a process in which the zona pellucida undergoes biochemical changes so that no additional sperm can actually penetrate. This prevents polyspermy, which is the fertilizing of an oocyte by more than one spermatozoon, and it results in embryo death. And in addition to the alteration of the zona pellucida, the cortical reaction is also believed to reduce the ability of the oocyte plasma membrane to fuse with additional sperm. Some species have a zona block as well as a vitelline block, and then some species just have one or the other. And so after membrane fusion, the nucleus of the spermatozoon is finally within the cytoplasm of the oocyte. The sperm nuclear membrane disappears, and the nucleus of the sperm begins to decondense. So decondensation of the sperm nucleus. Once the sperm actually enters and it's in the oocyte, like I said, the nucleus has to decondense so that that the chromosomes of the male pronucleus can pair up with the chromosomes of the female pronucleus. This decondensation of the sperm nucleus requires reduction of the many disulfide crosslinks that are formed during sperm maturation that was discussed earlier in spermatogenesis. So after these disulfide bonds reduction occurs, the sperm nuclear material or the male pronucleus is now available for interaction with the female nuclear material. And so the final step of fertilization is the fusion of the male and the female pronuclei, and this is referred to as syngamy. And then following this fusion, the newly formed zygote will enter the first stages of embryogenesis, which is going to be discussed at a later date. So to wrap it up, following sperm deposition, viable sperm that are not lost to retrograde transport or phagocytosis must 1. Transverse through the privileged pathways in the cervix. 2. They need to be transported through the uterus and the oviducts, where they have to undergo capacitation. They have to bind to the oocyte, undergo the acrosome reaction, and then penetrate the zona pellucida and fuse with the oocyte plasma membrane. And after the disulfide bond reduction occurs, the sperm nuclear material is then available for interaction, and the fertilizing male pronucleus is formed, which signifies successful fertilization. And I literally used the book, and I got all the pictures from the book, with the exception of one that has a source under it. So, thank you all for listening, and thank you all for participating, reading your side notes. Y'all have any questions? Yes, Mom.
You might have said it and I might have missed it, but is there something that keeps like multiple sperm from penetrating the zona?
Yes, I did mention it actually. It is the cortical reaction with those cortical granules that were released here. So whenever their contents are released into the preventative space, it actually makes the zona pellucida become, I guess, more solid, and it stops any other additional sperm from being able to penetrate it. Yes?
You mentioned the fertilizing sperm. How do we use that information to make decisions in terms of breeding?
So the further copulation is away from ovulation, the less likely the chance of actual fertilization taking place. So in animal reproduction, it's best for us to know when that ovulation is going to take place so that we can mate them or breed them in a corresponding time period. Yes, Erin?
You mentioned that for pigs, semen deposit in the cervix, whereas for like cows and even dogs, it's deposited in the vagina. Why is it different?
So it's different from pigs based off of their anatomy. I had mentioned that the cervix that I have here is for the cow and the ewe, but it looks different, which, as you mentioned, in different species. And so in the pig, inside of with the sow's uterus, it has these, I guess, like junctions that make or cause the pigs, what am I trying to say? That cause the pig's penis to be corkscrewed so that it can even fit through the cervix. So the anatomy of the cervix and of the male parts definitely will play a role in how the sperm is going to be deposited.
Okay, so this is actually like the membrane, the outer membrane of the cervix.
Yes.
Okay, so basically it tries. So how much sperm is like caught in that? So
I don't know if the exact numbers are known, but this is at the beginning, right? So it's at the cervix. So aside from the retrograde transport, where you're losing the semen, and aside from the phagocytosis that takes place in the vagina and in the uterus, this is another place where you're going to be losing sperm. And the semen. I'm not sure of the numbers. I don't think anybody really knows, because this is just a really understood phenomenon in reproduction. But yeah, a lot of sperm is lost here.
Can you just like run through? You already started it, but you just ran through like the major blocks of the sperm being to the oocyte. So I already said the phagocytosis, this thing.
Retrograde transport.
Okay.
It's blocked through the cervix because the cervix also acts as a plug, so it stops any bacteria or anything else that's entered into the female reproductive tract or copulation from entering into the uterus and everywhere else. But then there's also other blocks in every different place. So like I was saying when Erin asked her question, in the mare, I think it is, at the ambulary ischemic junction, they don't allow an egg that's not fertilized to pass through. So it's like there's different stops at each portion in the reproductive tract that aids in sperm loss.
Do you mind re-explaining this figure?
Yes. So in the cervix, there's two different types of mucus that is created. So this mucus here, the sulfomucins, is actually really, really thick, and it flows towards the vulva. So it flows out towards the exterior. Whereas you have the cialomucin that's really thin, that is flowing into the cervix. So as the sperm is traveling in the reproductive tract, it'll get caught in this thicker mucus, and that mucus is going to push it out of the reproductive tract. And the sperm that gets caught in the thicker mucus is actually allowed to pass through these privileged pathways.
So about the quick transport thing you were talking about, you go and ask the question, right?
Rapid transport?
Yeah, rapid transport. So what causes the rapid transport?
It's caused by the contractions of the muscularis.
So it's like as soon as the female is ejaculated in, I guess the muscularis is just working overtime.
So they say that, I guess, okay, when a female orgasms, I guess, her uterus and everything is contracting at the same time, and so that contracting is going to be pulling that semen directly up to it. Yes, Dr. B.
So you talked about zona and vitelline blocks. Do all species experience both blocks?
No, ma'am. Some species have both of them, like the cow, the horse, I believe. But then some species just have one or the other.
Is that picture associated with that?
There is. Oh, I don't know if there was a picture associated with the vitamin block, actually. But there is not. There is.
Okay.
But it would just essentially be, so the zona block is going to make the zona pellucida harden and stop the sperm from being able to come through. And then the vitelline block is going to make the oocyte membrane stop sperm that may have passed through the zona pellucida from coming through. And so it prevents polyspermy.
So that sperm in there is the one that's going to fertilize?
Yes, that's the one that's going to come through. Oh, vitamin. Okay, I'm sorry. I didn't hear you say that.
What are the programs again?
So their contents, once they are released through exocytosis, which basically just means that their plasma membrane is going to fuse with the plasma membrane of the oocyte, like you can see here, and then the contents that are inside of them, which is like proteases and ligases and stuff like that, it's going to be released, and then that is what actually hardens to make that's what actually makes the zona pellucida harden from the inside.
So like acne.
Yes. Any other questions? Yes, David?
How long does sperm stay alive if it has a weight population?
Good question. I actually have a picture and a table. I go over that. There we go. So, different species have different fertile periods, and the fertile lifespan of the spermatozoa is going to be different in each of them. So, in the dog, the sperm will stay fertile for whereas in the cow, it's only going to be fertile for 1.5 to 2 days. And so that's like I had mentioned that you have to have copulation and ovulation occur in a close period of time because, like I said before, if you're a woman and if sperm is deposited into you on Monday, that sperm is going to be viable until the next Sunday. So you have, you ovulate any time within that time period, you can become pregnant.
So that's why they take the Plan B, I guess. But
I guess so. I don't know what Plan B is, but I would assume that's why they take it. It probably kills the sperm.
Right.
No.
It makes your body think they're pregnant. So
that's not a part of my presentation, so I wasn't supposed to know that. Any other questions? Well, I just want to play my intro for y'all because there was only like 3 people here when I first got here. So let me just ask right quick, who has seen Look Who's Talking? Thank you, Dr. B. Okay, it came out in 89, so I know that's before all of us was born, but it's a really good movie. John Travolta, Bruce Willis. I don't understand why I didn't see this, but okay. It basically just shows how fertilization is.